Malashri, as Gustavo introduced me. I am a PhD student in the Tureki lab, and my project is on cell type specific multiomics of the dorsolateral prefrontal cortex in MDD. So first, I want to start with a little bit of background. As you all are probably aware, major depressive disorder is a serious psychiatric illness. It is characterized by persistently low mood, anhedonia, changes in sleep and appetite, and suicidal ideation, among many other symptoms. And depressive disorders affect around 200 to 300 million people worldwide, as per the Global Burden of Disease report from 2017. And according to the WHO, it is one of the leading causes of disability worldwide. And although treatment options with antidepressants are available, a lot of them are quite slow to act. And in cases of treatment-resistant depression, they are sometimes ineffective. So given the impact of this disease, it's unsurprising that a lot of biological and molecular studies have focused on trying to understand the basis and causes of this disorder. And for sure, a lot of rodent models have been developed, such as those using unpredictable chronic mild stress, social defeat, or maternal separation. And each of these models is useful for capturing some aspects of this complex human disorder. But as with any model system, there are also limitations, which is why many molecular studies have also focused on human participants using genetic, transcriptomic, and epigenetic technologies. For example, one of the recent GWAS meta-analyses identified over 100 genomic loci that are associated with MDD. There have also been transcriptomic and epigenetic studies using peripheral tissues such as blood, which have identified biomarkers of different subtypes of depression, as well as biomarkers of treatment response. Many of these studies have implicated peripheral inflammation and changes in the cortisol system in depression. In addition, there have been transcriptomic and epigenetic studies in the postmortem brain, which have found gene expression or DNA methylation differences in different brain regions between cases and controls, also identified sex-specific changes, and some of these changes were also specifically associated with early life adversity. The other thing to keep in mind is that the brain is a very heterogeneous organ, and it's composed of many different cell types. And a lot of these cell types have been implicated in MDD. For example, imbalance between excitatory and inhibitory neurotransmission has been associated with the disease, as well as changes in the number, distribution, and gene expression of astrocytes. Disruptions in white matter, thus implicating oligodendrocytes or oligodendrocyte precursor cells, have been reported, both from molecular and from neuroimaging studies. And in addition, some studies have implicated changes in the blood-brain barrier, as well as inflammatory changes in the brain. And of course, something that we have to keep in mind is that many of the earlier omics studies did not use a cell type specific approach because they weren't available at that time. So many of them relied on tissue homogenates. And when we perform sequencing studies of tissue homogenates, a lot of specific changes in a particular cell type can be masked. And a lot of the differences that we observe could be because of cell type composition rather than changes within specific cell types. And so in the field of neuroscience in general, and certainly for studying neurological and psychiatric disorders, there is currently a shift in focus towards cell type specific and single cell approaches for studying these diseases. So the first part of this presentation is about our attempt to use single nucleus RNA sequencing in the DLPFC of a cohort of male subjects with MDD. This cohort consisted of 17 psychiatrically healthy controls and 17 depressed cases who died by suicide. And we used tissue from BA9. We used the 10x Genomics Chromium platform for high throughput capture of single nuclei, followed by library preparation and sequencing. And this is a droplet-based approach, as you can see over here. We use standard analysis approaches using tools like CellRanger and Surot to perform clustering and differential expression analysis, followed by other bioinformatic analysis. And we also did some validation experiments, although I'm not going to present them for this presentation because we have newer data that I would like to show to you today. And so the unsupervised graph-based clustering gave us 26 different cell type or cell state clusters within our data. We found that there were more neuronal cells than glial cells, and this was in line with what had been reported at that time from snRNA-seq studies of the human postmortem brain. And we also found that the majority of cells belong to excitatory neuronal classes. We investigated the relationship between the different cell types that we identified, and we also examined the expression of marker genes and found the expected patterns. For example, genes such as SNAP25 or RBFOX3, which are pan-neuronal, were expressed in both inhibitory and excitatory neurons. Something like SLC17A7, which encodes the vesicular glutamate transporter, was enriched in the excitatory neurons. GAD1 and GAD2 enzymes, which synthesize GABA, were enriched in the inhibitory neurons. Similarly, myelin proteins like PLP1, MAG, MOG, or MBP were enriched in oligodendrocytes. PDGFRA or OLIG1 and OLIG2, which are known markers of OPCs, were mostly enriched in the OPCs. Astrocytic markers like GLUL or NDRG2 were enriched in astrocytes. And in addition, we were also able to identify CLDN5 expressing endothelial cells and microglia marked by CX3CR1 expression. Interestingly, we found that the average number of UMIs was lower in the glial clusters compared to the neuronal clusters. And at that point in the literature, this was again consistent with what other groups had found. To further understand what are the different excitatory neuronal clusters represented, we looked at the expression of a set of marker genes that are known to have a gradient of expression across the different cortical layers. So starting with CUX2 or RAS-GRF or PVRL3, which are enriched in upper layers, all the way up to SYNPR or TLE4 or NTNG2, which are enriched in the deeper layers of the cortex. And in fact, we found that there is a gradient of expression of these layer-specific markers across our different excitatory neuronal clusters. Similarly, for inhibitory neurons, we found specific expression of the mutually exclusive markers such as SST, VIP, and PVALB, which would be expected in inhibitory neurons of the cortex. For our oligodendrocyte lineage cells, we performed pseudotime trajectory analysis and found that OPC2 represented the most immature oligodendrocyte precursor cells in our data, followed by OPC1. And then oligo2, oligo3, and oligo1 represented oligo1, which represented the most mature, let's say, myelinating oligodendrocytes. We found that the expression of genes that are expected to vary across the oligodendrocyte lineage showed the expected patterns. For example, MAG, MBP, or MOG, or PLP showed a gradual increase in expression with pseudotime, whereas genes like PCDH15 or PDGFRA showed a gradual decrease in expression across pseudotime. Another kind of interesting finding was that when we looked at the genes associated with pseudotime in the cases and controls separately, and we focused specifically on the genes that were only associated with pseudotime in the cases or only associated with pseudotime in the controls, we found that these 2010 genes actually showed an enrichment for the apoptotic pathway. In addition, we performed deconvolution analysis of our oligodendrocyte clusters and compared them to the Jekyll et al. 2019 dataset. And we found, as expected, that the OPCs showed strong correspondence to their OPC cluster, as well as the oligodendrocytes primarily corresponded to their oligodendrocytic clusters. Interestingly, this oligos 3 cluster showed a small correspondence to the immune oligodendroglial cluster, which they identified as oligodendroglial cells that had some expression of immune markers. And this is quite interesting because in our t-SNE plot, which is the dimensional anti-reduction showing our different clusters, we find that there's a small population of microglia that seem to be right adjacent to a small population of the oligos 3. And we think that this is probably driven by these immune oligodendroglial cells, which may be present in this oligos 3 population. So at this point, we felt confident that the clusters that we had defined made biological sense. And we decided to perform differential expression analysis using a mixed effects linear model and accounting for the number of UMIs per cell, which accounts for library size. and also for batch and subject. We only included genes expressed in at least 10% of the cells, and we included cells that had a high correlation with the median expression profile of the cluster. And so this is a summary of our differential expression results. We found that there were 96 cell type specific differentially expressed genes, 80 of them, so the majority showed downregulation, whereas 16 showed upregulation. In this plot, every single circle represents one of the upregulated or downregulated genes. They are colored according to the level of significance, and the distance from the midline represents the extent of up or down regulation observed. In particular, the OPC2 cluster and the excitatory 7 cluster had the highest number of differentially expressed genes. We also found that the number of differentially expressed genes did not necessarily correspond to the cluster size, which was reassuring. So the cluster size in this plot is represented in the size of the circle, because if we were finding more differentially expressed genes purely based in how big the cluster was, then that would be a bit more concerning. So these were our differential expression results, and in particular, OPC2 and excitatory 7 seemed like interesting targets to follow up on. We also examined whether there was some correspondence between our differentially expressed genes and what has previously been reported in the literature for MDD. So we looked at the overlap of our genes with the SciGenet dataset or database, which performs or which contains gene disease associations based on text mining of a large number of publications. And we found that the most associations were found for MDD, followed by schizophrenia, with low levels of association for alcohol use disorder, bipolar disorder, or cocaine use disorder. We also looked at the DisGenet database, which is a larger database of disease gene associations. And we looked particularly at a set of terms associated with depressive disorders. And we found that 15 of our differentially expressed genes were linked to these terms in that database. And this was a statistically significant overlap. Unsurprisingly, the main contributors were OPC2 and excitatory 7, which had the highest number of DEGs to begin with. We also performed as a complementary approach weighted gene co-expression network analysis, or WGCNA, by taking the average expression per subject across all the clusters and cell types. We found that the largest module associated with depression was the blue module, and there was a significant overlap of 41 DEGs with the blue module. In addition, when we performed hub gene analysis, there was again a significant overlap of the differentially expressed genes with the hub genes for the blue module. And in particular, 15 of the differentially expressed genes from the excitatory 7 cluster overlapped with these 41 genes, and 3 genes from OPC2. We also included in this WGCNA analysis the percentage of nuclei in each cluster as a covariate. And excitatory 7 was one of very few clusters that was associated with 4 out of 5 of the modules that were linked to depression. There was also astrocyte 2, which was associated with 4 out of 5 of the modules, but it did not have any differentially expressed genes. So based on both these approaches, the differential expression analysis and the WGCNA, we felt that excitatory 7 could be a promising target for follow-up. So we decided to look at how we might isolate cell types corresponding to this excitatory 7 cluster for DNA methylation comparisons between cases and controls. So first of all, we needed to know what was defining excitatory 7. And when I looked at the different markers specific to subclusters of excitatory neurons, I found actually that excitatory 6 and 7 were of relatively similar expression in a lot of these markers. And in particular, because we wanted to sort nuclei, I wanted to look for proteins that would be present in the nucleus, which we could target using antibodies. So I focused on transcription factors that were enriched in excitatory 6 and 7. And I found a couple of transcription factors that were common to both of them. As you can see here, statistically, RORB and FOXP2 were enriched in both of these clusters, and those were the only 2 clusters showing enriched expression of both these markers. And I decided, instead of focusing on the one transcription factor enriched in excitatory 7, I decided to focus on the 2 that were common to both of these clusters, because even excitatory 6 showed some differential expression, and actually one of the differentially expressed genes in excitatory 6 overlapped with those differentially expressed in excitatory 7. I also wanted to ensure that this excitatory 7-6 cell type was not just an artifact of whatever clustering approaches I used. And so I performed deconvolution analysis to compare my data set to the Mathis et al. Data set of the prefrontal cortex containing control subjects and Alzheimer's patients, as well as the Velmishev data set, which looked at individuals with ASD and controls. And I found that my excitatory 6 cluster corresponded mostly to excitatory 5 in the Mathis dataset, and excitatory 7 corresponded mostly to excitatory 1. And when I looked at the expression or enriched expression of RORB and FOXP2, in fact, excitatory 5 and excitatory 1 both showed specific enrichment of RORB and FOXP2. There was an additional cluster, excitatory 7, which also showed enrichment of this marker. But since none of my cell types corresponded strongly to this cluster, I felt that RORB and FOXP2 would be sufficient for isolating my cell type of interest. Similarly, in the Velmishev dataset, both my cell types, excitatory 6 and 7, actually corresponded to a cell type that they broadly annotated as layer 4. And that was the only cluster in the dataset that showed enrichment for both FOXP2 and RORB. So confident that it made sense to target these genes or the proteins encoded by these genes, I spent several months optimizing a fan sorting protocol using RORB and FOXP2 antibodies. And I found a set of conditions that were sufficient for replicably isolating a population of cells high in both FOXP2 and RORB. So on the x-axis is the RORB signal measured by FITC, and on the y-axis is the FOXP2 signal measured by Alexa 647. And as you can see, each of these three subjects has this population over here, which is high expression of both RORB and FOXP2, which is the cell type which is the population of cells or nuclei we would like to validate. So to confirm that the gene expression in this population actually corresponds to excitatory 6 and 7, we will be performing qPCR as well as some sequencing experiments performed by followed by deconvolution analysis. And once we are confident in our sorting, we will perform enzymatic methylation sequencing on the sorted nuclei to compare DNA methylation profiles between cases and controls. The last part of this presentation is about extending the cohort that we have with a number of female subjects. And the rationale for this is, of course, in any case, biological research needs to include more female subjects because females are understudied. But in the context of MDD in particular, we know that there are differences between men and women in the rates with which they are diagnosed with MDD, as well as the rates at which they die by suicide. And there are social and biological factors that contribute to these differences. In addition, there's evidence from molecular and behavioral studies in humans and animal models of differences between men and women, or males and females, in terms of MDD. So this is the assessment of cell types in our female data set. The expected patterns for neurons, non-neuronal cell types were observed as we saw previously for the male data set. And when I compared the unsupervised clustering annotations here on the x-axis with the predicted labels from the male data set, in many cases, I saw a good one-to-one correspondence between the cell types that were identified from these two approaches. In particular, the excitatory 7 cell type that we're interested in corresponds to excitatory 4 in the female data set, and the excitatory 6 cell type corresponds mostly to excitatory 9 and 12. And we also wanted to further explore the association of these cell type signatures with different spatial locations. And for this, we leveraged the Visium dataset recently produced by Maynard et al. In the DLPFC. So this is one single female reference subject that we used for this purpose. And we found, in fact, that the excitatory 4 cell type signature was mostly enriched in the deeper layers of the cortex, as one would expect. The excitatory 1 cell type, in contrast, which is supposed to be upper layer, had its signature mostly enriched in the outer layers of the cortex closer to the edge over here. Similarly, excitatory 9 showed a stronger signature in the deeper layers of the cortex, although there was some enrichment in the upper layers as well. And as a representative glial cell type oligodendrocyte 1 showed a very strong enrichment in the white matter, as one would expect. We performed pseudotime trajectory analysis as well with these data and found the expected gradient from OPCs to oligodendrocytes, and we recapitulated the expected increase in expression of myelin-associated genes and decrease in expression of genes enriched in OPCs. So at this point, the next steps for us would be to perform differential expression analysis within each cluster to compare cases and controls, and of course, to compare these results to our previous results from the male cohort. We also want to analyze both the male and female data using the same pipeline for a combined analysis. And eventually, the goal is to identify targets for follow-up, potentially even using animal models. And so these are some people I would like to thank for all of their contributions to this project, Volodymyr and Daniel for a lot of advice and help with the fan sorting, Matthew Suderman at the University of Bristol, as well as JF, one of our bioinformaticians, for all of their advice and help regarding the data analysis, the Ragusa's lab for processing a lot of the male samples for library prep, and Deborah Mash's lab for providing some of the female control subjects, as well as my supervisors and my committee members for all of their inputs and guidance.
Very good. Thank you, Mallory. Maybe you could stop sharing so that people can see each other, and we have time for maybe a few questions. Yes.
I mean, I think that's very cool, and I don't know. I mean, I'm sure you're aware of the paper from Eric Nessler where they looked at bulk RNA sequencing in major depression stratified by sex also, and they saw almost no overlapping shared loci, which is very interesting. So doing that on a cell type specific way will be really interesting to see, like how sex specific molecular differences actually extend to the cell type level.
So I think that is definitely our goal, and we're very interested in that. The only limitation is that because of the way our experiments were designed, all of our female subjects are in the newer cohort and all of our male subjects are in the previous cohort. So we will have to come up with some kind of clever meta-analysis approach to combine, but that's what we hope to do.
And there was also a technical issue, isn't it, as well, Mallory, because some of the chemistries that we use changed.
Yes,
but in,
yes. So
you know, it's just that the way things evolve change, and that brings additional challenges. Yes.
Yes, because all the older subjects are with version 2 chemistry, and the newer subjects are both with version 2 and version 3 chemistry from the Chromium system.
Okay, so if you really wanted to do that, you would need to resequence, like basically generate a new experiment.
Yes, and even make the libraries again, because the actual capture technology is different.
Yeah, but we are hoping to find analytical solutions to deal with that.
Yeah.
Mallory, thanks for your talk. I wanted to ask you about the Visium analysis you did. So you didn't generate these data, as I understand it. So how's because we haven't used it yet, and we've kind of wondered whether it's useful for us in our system. How do you find in terms of specificity to a single cell? How do the tools for deconvolution, because you capture up to 10 cells from my understanding, depending like in the brain, right? So how does that work for you? How do you find it?
So I think the resolution is now down to like 3 or 4 cells potentially. I'm not the expert though, but I find that the signatures are definitely not as specific. So when I did deconvolution with other single cell data sets, there was really in many cases a 1:1 correspondence. Whereas with the Visium data, it's kind of a range of spatial locations that's enriched for a particular cell type. And I did find that, for example, for astrocytes, they somehow show a specific enrichment in layer 1 and 2, which is not what one would expect. But I think, so I only used kind of label transfer based on SURAD, which is kind of only one method for assessing this, but there seems to be other methods such as the RCTD, I think robust cell type deconvolution, which is designed for spatial data, for deconvoluting spatial data. So I hope that those more specific methods will give better resolution essentially for determining that.
But do you have to apply, so would it be helpful, let's say, to have data, single nucleus dissociated and a Visium data from the same samples processed in your own system, let's say, to improve the deconvolution? So in your case, I guess that's not the case because they're coming from different sources.
Yes, but it really depends on what you would like to do and how detailed your Visium data is. For example, I know that it's possible to use antibodies to look at specific cell types. So if you have a cell type in mind and you use antibodies to mark those cell types and the spatial spots that contain those cell types, then I think it could give you an edge when you're trying to determine which cell types are in which spots, essentially. But we don't have that data yet to work with, so I don't have practical experience with that. I'm very new to it, so I don't have a more informed answer, unfortunately.
Marina?
Yeah, I can make a few comments on it. So my first comment would be to wait because they're coming out with a much denser array for the spatial work, and they're claiming it should be single cell. Whether or not that's true is, but anyways, within the next year, they're supposed to come out with this. The other thing I can say is that one way, and something that we were discussing recently, because we do have a few projects going on trying to work with Visium, is some of the annotation will be helped by people who are very good at histology. So you can sort of annotate the sections to give a better idea of what cell types you think would be there. But that still will, the genes that are associated with a given spot can still be muddled, but you would at least have, if, for example, you have, regions that you would expect. So for example, the image that Mallory shows from the Maynard data is first annotated manually. And so then you can reliably know that sort of this is where one region of the cortex would end and another would start, and you could be more confident that you would get a certain cell type. So some of those sort of pre-manual annotations would help in terms of classifying your cell types. But also, like I said, they're coming out with more resolution. So I would actually wait before trying to jump into it, because I think you might get something more useful in the future.
Thanks for the advice. Yeah, I mean, for us, it's an additional complication you'll see in my talk. I mean, we're, especially when we're dealing with brain organoids, which are not, so the annotation at the histology level becomes much more complex.
Right.
And unreliable. So yeah, but I'm interested to see your experience with it.
Yeah. It is my understanding that the Maynard group, so are you familiar with this paper from the Liebert Institute where Mallory got the data?
I mean, yeah, but I didn't read it in detail. So.
Okay. So it is my understanding that they do use the combination of the droplet-based single cell with the spatial, but it still has its limitations, because you're still going to have that lack of resolution with the Visium. So the most resolution I've seen is coming from these sort of Cartana probe-based single cell. Do you know what I'm talking about?
No, what's this?
So these are using sort of like a MERFISH. It uses a probe so that it's not as high throughput. You don't get the full transcriptome, but you can get up to 100 different genes, but you're definitely getting that single cell resolution. So I don't know if that's useful, but if you really, really need the resolution, those seem to be the better. They're called in situ sequencing techniques. And they seem to have the much higher resolution, much more confidence in the resolutions that way.
Cool, thank you.
Okay, any other questions? No, thank you, Malasri. Elizabeth, you want to?
Yeah, so we we have another from our lab, Christiana, which I think some of you may still know personally. Definitely Gustavo and Nagi. She will present work on how we try to use brain cerebral organoids to look at like prenatal stress models. And combining also single cell techniques, FACS, and all other things, just so that you get an idea of the types of experiments that they're doing in the lab. So Christiana, do you want to get started?
Hi, yeah, so it's really nice for me to present today. As Elizabeth mentioned, in my previous life, I was there, so it's really nice to see some of my friends on this screen. It's been a while since I've been to Montreal, so at least we see each other on the screen. Yeah, so I've thought of this presentation in a different way. I'm getting a heart. That's very sweet. Thanks, Jermaine. So I've thought of this presentation in a different way than Malasri. So let's see if I manage to generate a little bit of discussion. So I won't be going into too much detail about particular findings from one story, but rather I'd like to give you an overview of the different projects we've conceived in this space of trying to understand prenatal stress, prenatal exposures, and using in vitro models. And in this case, I'll focus on cerebral organoids as the model of interest. So feel free to stop me along the way if there's something you find particularly interesting, or we can see if there's time for questions at the end.
Yeah, Christiana, you're not in presentation mode. I'm not sure you know that.
I will do this. All right. So in the Binder lab and relevant to this particular initiative, we're very interested in understanding sort of the embedding of genetics and environment in how psychiatric disorders are developed, and we want to look at that both from the level of the cells and as high as the level of populations. And so we are, here we go, particularly interested in mental disorders and how they come about. So we know that they are a combination of genetic susceptibility as well as epigenetic changes that are instilled by environmental exposure. So the topic of this initiative is very relevant here because we know from previous research, including from both of these labs, that early life experiences can lead to or exacerbate genetic susceptibility to lead to mental illness presentation later in life. And particularly even as early as the prenatal period, these environmental exposures in combination with genetic susceptibility can play an important role in lifetime susceptibility so as risk as well as resilience. So we're trying to understand these mechanisms in a way that's sort of comprehensive in this very complex way. We're particularly interested in stress and more specifically how stress is mediated by glucocorticoids exposure. So glucocorticoids being primarily endogenous cortisol, as well as synthetic glucocorticoids like dexamethasone and betamethasone, which are given during pregnancy, for example, for speeding up maturation of the fetus, but have also been linked with neurodevelopmental and neuropsychiatric outcomes later in life. So glucocorticoids, just a brief reminder, are steroid hormones that act systemically and respond to the environment. They modulate stress via the HPA axis. And in the fetal environment, we know that there's a possibility of transmission from the mother. So if the mother has elevated glucocorticoids from stress or other exposures, these could pass through to the fetus via the placenta. And there are mechanisms in place to control this, but these mechanisms could also be impacted by the environment. At the cellular level, we know that glucocorticoids play a role in regulating gene expression via the glucocorticoid receptor primarily and the mineralocorticoid receptor. And as a transcription factor, the GR can then impact the expression of many genes, potentially in cell type specific ways, activating different pathways. So we're really trying to understand these mechanisms. And importantly, it's been shown, including by our lab, that these effects can be lasting. So these cellular responses can potentially be imprinted, for example, in epigenetic ways that can last and lead to vulnerability later on. So in this context, I'll talk about two areas of projects that we're currently working on. One, about understanding how these exposures during these very plastic periods of development can lead to potential remodeling or shifts in the way the brain is developing. And secondly, more at the cell type, single cell resolution, to understand pathways that are activated by these exposures and how these interact with each other, potentially leading to lasting effects. So how do we study an active process that's going on in utero and the molecular processes involved to impact the developing brain when we don't have access to this tissue? So of course there are animal models, but given that neuropsychiatric traits are very complex, not all aspects of these phenotypes can be modeled in rodent models. And access to non-human primates is limited, so we're kind of left to piece together different aspects of different tools that we have access to. So through peripheral samples in cohorts of human patients, postmortem brain, as Mallory already discussed, in our case, looking at perinatal tissues like placenta and cord blood, and adding in the mix models that we can manipulate, like in vitro systems. And we think this sort of combinatorial approach can lead to a way to study these active processes and the molecular mechanisms involved. So I'll focus my talk on our use of cerebral organoids in this area. And so I'll just introduce them briefly. So they are in vitro systems based on or derived from induced pluripotent stem cells. They are three-dimensional, so they look like this little ball of cells. They're derived in vitro, so we have incubators full of them, and when we do immunohistochemistry staining for them, they look something like this. So they replicate the functional unit of the human brain, which is this ventricle, and they show the same kind of developmental pattern where you have the immature cells on the inside here, and as they mature and they get they migrate upwards into the outside of the ventricle. So one of the first things when we started working on these projects now a few years ago, we wanted to make sure we characterize them properly to know what we're looking at. So just keep in mind, this is a tool, we're not working with actual brains, so there will be limitations to this tool. We're hoping the benefits that we gain from them beyond what's already available can help us understand the things that we couldn't understand before. So the first project I'll briefly mention is one where we looked at the developmental trajectory of these organoids from 17 days in culture to 158, so following about 4 months, to understand sort of how they develop on a linear or temporal trajectory and how similar they might be to the brain. So we did bulk RNA sequencing at the time. So one of the things I'll show you, so if we look at this developmental pattern across these 7 time points, we see progenitor genes, so genes defining progenitor cell types kind of coming up and more abundant, and genes that represent neuronal cell types coming up as a little bit later, but also being expressed sort of consistently, suggesting that the different cell types from more mature to more immature to more mature are constantly present, just their proportions probably altered. The other thing we did here was to see how at the bulk level these organoids fit with other data sets that have profiled expression in the brain or in vitro systems. So here in this PCA, what you see is bulk RNA sequencing from different sources. For example, here you have induced pluripotent stem cells. As a control, we have blood from GTEx, and then as you see, this sort of data sets fall nicely on a linear trajectory where the in vitro systems fall on the lower end. And then we go into prenatal brain from the BrainSpan data set, which is bulk RNA-seq, and into more mature up to adult bulk RNA sequencing data sets. And when we look for our organoids, I just put this arrow here because the dots are not so visible, but they follow this linear trajectory based on their time in culture. So they don't follow the trajectory or the timeline specifically to the day. So 4 months in vitro does not equal 4 months in utero, but they do follow the same trajectory. And the older organoids definitely fall within the fetal brain sample. So that's encouraging to see that they follow the transcriptional patterns of the tissue we're trying to model. Yes, the other thing here, this is a sort of early work in progress. Now there's a couple of data sets of fetal brain available at the single cell level. And since we're also doing a lot of single cell approaches, Leander, a PhD student in our lab, is trying to integrate these data sets to try to see if we can gain more information from our single cell data in looking at how they integrate with the fetal. So here Leander took 3 of our data sets. They're called Binder and then the cell line. And then this is the data set. The first author is Poliodakis, last author is Geschwind. So this data set is week 18 in utero, so about mid-gestation. So what you can see here is the colors, so representing each data set, are pretty distinct still, but there is a fair level of integration. But if we separate the two, and here we're looking at the cell type labels that they've given, where here you have the more mature excitatory neurons, inhibitory neurons over here, and then their radial glia, which are the progenitors of the developing brain, are over here. And if you look at our data sets, in orange you have our neurons, and you see that they follow nicely along the same space as the data from the Geschwind data set. What we have in organoids, for example, this in green, we have a lot of what we've called non-neural progenitors, which are progenitors of other cell types, mostly mesenchymal, stromal, that are supportive cell types that keep this 3D structure together. And some of them are recapitulated in human brain as well. But when we're looking at these data sets where they've dissected specific areas of the cortex, of course, you're not going to find them. So this highlights the importance when we're working with these systems to look at the single cell level or cell type specificity, so we're not confounding what we find to cell types that are not relevant for the brain. The other thing it's also telling me is that there is fairly good overlap with the fetal brain, so we're not just looking at fibroblasts. So okay. Yeah, and the other message I would say is in terms of the maturity. So probably if we were to look at more specialized cell types, we would be able to recapitulate at these cell types that are more mature at the total brain level. Okay, so I'll give a sort of brief overview of the different paradigms we're using in this system. So we take organoids in vitro and we want to understand the effect of glucocorticoids on them. And for glucocorticoid exposure, we're using dexamethasone, mostly at a concentration of 100 nanomolar, but we've also tried different other things. And we're doing exposures that are acute, in this case 12 hours, and I'll label them with this little star here, or chronic, which is quite clear here. So one of the first projects we did was to do 3 different acute exposures at 3 different time points in the organoid's development to understand if temporal or cell type specific differences exist between the response to glucocorticoids. And in this story, we've done single cell RNA sequencing, and yeah, it's mostly finished, but I won't go into too many details about it. Then the other projects we're working on, where we're using a more complex design, where we're either doing a chronic exposure and then assessing the response immediately, or we are stopping the treatment, doing a washout period of 20 days, and then either assessing the response or doing a second hit of an acute exposure. So then to understand this susceptibility and vulnerability or resilience that could be a response to the glucocorticoid exposure early on. And in this story, we are doing single cell RNA sequencing as well as single cell attack-seq to look at chromatin accessibility and to understand the dynamic between these 2 levels of regulation. So how the open chromatin regulates gene expression and how dynamic open chromatin is in this context. And finally, because most of the research looking at epigenetic changes as a result to stress exposure, for example, especially in utero, has been done at the level of DNA methylation, which we know to be responsive to elicit lasting changes that can then regulate gene expression. So we cannot do this, or we could, but it's not very lucrative to do it at the single cell level. So instead, we're using FACS sorting to sort two different populations of progenitors and neurons, sort of the major populations in this, and then doing whole genome bisulfite sequencing to understand the cell type specific DNA methylation response. So just a couple of findings from the acute exposure project. So here we find different cell types that are relevant in the developing brain context. I won't focus too much on it. We do find rather quickly that they cluster mostly into neurons, neural progenitors, and these non-neural progenitors that we find have quite a different response to glucocorticoids, and in fact, quite a bit stronger than the neuronal cell types. And so it's important to look into them. And then looking at just the kind of the genes from the different cell types. Here I've just grouped in the three major cell classes. So some of the genes that they share are things like PAX6, SINE2, TCF4. These are all genes that have been associated with psychiatric disorders and neurodevelopmental disorders, and they're as well as being important in brain development. And when we take the genes that are differentially expressed between treated cells and untreated cells in these three major cell classes, and we do an enrichment analysis in different data sets of associated to disorders, for example. So the first thing I'm showing here is the cross disorders GWAS, including the major psychiatric disorders that was published recently last year. So when we take the genes that are GWAS genome-wide significant in the cross disorder analyses, and we ask, is there an enrichment of our differentially expressed genes? What's interesting is that even though there's a lot of differentially expressed genes in the non-neural progenitors, in fact, the larger group, there's no significant enrichment with the psychiatric disorders related genes. The neural progenitors are sort of almost significant and quite a strong enrichment in the neurons. We do the same, we ask the same question with genes associated to intellectual disability, and we find something very similar, strong enrichment for neurons. So we think the more specialized cell types, though perhaps their fold change in terms of response to glucocorticoids is not so strong, it does seem to be more specialized to these disease-related genes. So if we see that this acute stimulation can elicit an immediate transcriptome response from genes related to psychiatric disorders, what would happen in the long term? So that's a question we're asking. So would there be lasting effects of this acute dexamethasone exposure, or would you have to really do a very long chronic exposure to really have the long-term effects? Is this something that would just go away, you know, this overstimulation or understimulation? So by qPCR, the first things we wanted to see is there any lasting effect in these organoids, period. So we did another 12-hour treatment. We collected organoids bulk and we did qPCR. This is a gene FKBP5 that our lab has studied a lot. So over the course of these things, you'll hear a lot about it, but it's a gene that's well known to be a responder to glucocorticoids. It never fails. So we see a very strong response immediately. What's interesting is 2 days, 6 days later, we still see a significant upregulation. So these effects are lasting, at least in the genes that are strong responders. Here I'm showing PAX6, which is a gene that's important for neuroprogenitors. And here as well, we see a lasting effect, though not as consistent and not as strong of an effect, but certainly notable. So this here is a picture of the organoid structure. And so these ventricles that I was showing you in the immunohistochemistry pictures. So if I think of this as a ventricle, which is the same in the human brain, basically you have the progenitors down here. So these would be the PAX6 positive cells. And as these cells are maturing, they start migrating upwards into upper layer neurons, and then the deep layer neurons follow. And here are some of the markers. So SATB2 would be upper layer neurons, CTIP2, TBR1 in the order of their development. So if we are seeing an upregulation in PAX6 or during this period, if there's a lasting effect to it, we would expect that if we're increasing the number of these cells a few days later, that we would have more of those newborn neurons that would be the result. And if that's true, then that could change the brain architecture and have a lasting effect. So what we wanted to do is see if that's possible. So we did a lot of staining of organoids and counted cells with these 3 markers of different layers. Now, the layer organization in organoids is not so nicely defined as in the brain, but you do see the cell types kind of coming up in the same order. So we counted cells only in the ventricular zone. So even though you see a lot of CT2 positive cells here, what's more interesting to us are the ones here that are early on, that are basically the new formed neurons that are still in the process of migrating. So when we counted this, SATB2, which is the more mature marker here, there were very few cells in the ventricular zone, which means that there weren't so many new cells that were made. Quite interestingly, CTIP2, there was a very significant increase in the number of new neurons of this particular type, and TBR1 was not as strong of an effect. And given that the age of organoids we did this in, which was 60 days, this makes a lot of sense because the CTIP2 cells would be the ones that would be sort of newly made at this time. So we didn't make new cell types, but what we did is we increased the number of the cell types that should have been there. So now we postulate that if this exposure had been even longer, let's say a chronic exposure, and this process was lasting, then you could potentially significantly alter the direction or the architecture of this structure. So with that in mind, I'll move on to some of our chronic projects. So this idea of can we shift the architecture is very interesting, and it could make a lot of sense in terms of the outcomes that could happen as a result of exposures in the prenatal period, as for example, neurodevelopmental disorders, intellectual disabilities. So one of the things we wanted to ask then, do these potentially have effects on lineage, neuronal lineage? So what kind of neurons these progenitors can become? So this is using single cell RNA velocity, which is a tool that basically uses the proportion between spliced and unspliced transcriptome to understand the dynamics of what's newly made and what's on the way to being done. Am I okay for time? I see that it's 5. Can I continue?
Yeah, I'm not sure. Well, I just discussed quickly with Jermaine, and she said that it's probably okay, but we could get a feedback. I don't know.
I think it's okay on our end as well.
Yeah, it shouldn't be too long. I mean,
No.
just a few more minutes, exactly. Yeah.
All right. Yeah, so we're doing RNA velocity to understand lineages, shifts based, and to understand how this is different between control, so what the cells are supposed to become, and when we give chronic dexamethasone. So I think maybe you can appreciate from these heat maps that we see some difference in abundance of these cells on this lineage. And so now we're trying to figure out ways to how to compute these differences between them. So, and the other thing we can do when we're trying to understand these differences in lineages is look at the open chromatin, because this is showing what is being poised for expression and thus important for the next stage, what the cells will become. So we're doing the same with single cell ATAC-seq. So here I'm just showing some data where we did, again, a developmental trajectory, just using day 30, 45, 60, 70, and 90 organoids. We can cluster them based on transcription factor start sites or other features of the genome and get more or less the same clusters we do with the transcriptome. And here we can look at peaks where so this is wild type, there's no treatment here, but basically you can see peaks. So this is a gene that I mentioned already, PAX6. So if we look at PAX6, where are the peaks where the chromatin is open and what parts of the genome is it interacting with in this ATAC-seq data, and what cell types it's more specific in. We can do the same with a more neuronal marker, DCX, where here we see much more specialized based on the cell types that should express it. And then when we apply this approach to our treated and untreated cells, we can compute differences between these peaks to see if things are closing too early or opening sooner, opening too much, and so on and so forth. And finally, in the same paradigm, then we want to add DNA methylation. So like I mentioned, we have this paradigm where we're doing a chronic exposure followed by an acute stimulation, and we want to understand the interaction between the epigenome and the transcriptome. So we've done FACS sorting using DCX for neurons and PAX6 for neural progenitors. And actually, we're doing this project in collaboration with the Mini Lab, specifically Nico Tool and T1, whom you guys, I think, know. And so we're trying to understand which loci are dynamically responding to the stimulation, whether there are lasting effects of this treatment that we see even after the washout period, and whether some loci respond to having the history of the chronic treatment. And so the goal here is to basically overlay this information, the attack-seq, to see where the chromatin is open, where the DNA methylation is reactive or there are lasting changes, and finally to understand how that translates to the transcriptome. So going back to, this slide is basically to say that, so of course this is interesting, but it's all in vitro and it's only showing us interactions at a molecular level. So in order to complement that, we're working on collaboration with the University of Helsinki, primarily Katri Raikkonen, and in the lab, Darina and I are working on this project to try to understand the early exposures during pregnancy by following mothers throughout the pregnancy, as well as their children after birth. And we have access to perinatal tissues like placenta, cord blood, as well as early placentas from the first trimester, from chorionic villus sampling. And we've done DNA methylation, RNA sequencing in these data, as well as looked at diurnal cortisol. So we're really trying to understand how the exposures during pregnancy then can lead to outcomes in the children, be it behavioral or developmental. And so that'll probably be a project that's a topic of one of these presentations. Okay, so finally, just a few outstanding questions. So, I put together this idea about how we can model these processes in vitro and using organoids as one of the tools. But of course, there's still some things that we're not able to model that I think are outstanding questions that are interesting for this group. So for us, particularly how the placenta mediates the transmission of glucocorticoids from the mother. So we're not able to control in this system that aspect of it. Not too much is known about it either. How the blood-brain barrier then mediates the entry of glucocorticoids. This is another level of control that we're not able to model. And then what are critical developmental periods of exposure? Because as you saw with the staining and counting experiment, that really depends on when you're looking to see what effects you're having on what types of neurons. And finally, what are actual concrete outcomes of these exposures in terms of risk versus resilience? So with our models, I think we can build a sort of a hypothesis for what are the pathways that are being activated. But of course, it's really hard to say whether these complex exposures in a human really translate accurately in vitro, in a Petri dish. So that's my last slide. I just want to thank everybody in the lab. So Elizabeth Binger and Silvia Capello from the Institute. And I work with 3 really great PhD students, Antti, Leander, and Leah, who have contributed to a lot of these studies, and then a lot of our collaborators here at the Institute, and of course the Mini Lab and the Raikkonen group from, as well as Fabian Theis from the Helmholtz Institute. So I can stop here. I don't want to push it too far. I don't know if we have time for questions. I'd love to hear your impressions. So I'll stop sharing.
Thanks, Christiana.
Yeah, sorry, I rushed a bit in the end there.
Christiana.
Yes.
Nagib.
Hello, Nagib.
Hello. Nice presentation.
Thanks.
Nice data. I was just wondering, so talking about the cortical architecture, cytoarchitecture, so it seems established and quite clear that there's radial migration of principal cells. What about interneurons that mostly migrate tangentially? Is there evidence of that in organoids?
Yeah, so in these organoids, we're using a protocol that doesn't, there's no patterning. So basically it doesn't drive them in a particular direction, and we tend to mostly get dorsal. which means we don't get a lot of interneurons in these, but they can happen. So we get mostly precursors to excitatory neurons, but I don't know of any study that's specifically looked at that. Antti would be the person to have looked more detail into that. So I don't study a lot of migration stuff, but Antti's looking into it in maybe you should, if she's here. Yeah, go ahead.
So yeah, we're using an unpatterned protocol for organoids. So theoretically, we would be able to get both. We get mostly dorsal cortical ventricles, but we do have also ventral ventricles in the same organoids. And recently also I find double ventricles. So half the ventricle is dorsal, half is ventral, which is actually very interesting on the migration aspect. There are ventral patterned organoids that we can do, and in our department, there is a project that fuses ventral organoid with a dorsal organoid, so then you can study migration patterns of both dorsal cortical neurons and ventral tangentially migrated neurons. And there is a migration between the organoids using that protocol. So all is possible, though with our unpatterned organoids, since we get mostly dorsal cortical organoids, the number of ventral cells is very little to see effects there, I would say. So I think we would need to pattern to see effects there.
Interesting.
Yeah. And also it's not something we've looked into, but so the group of Silvia Capello is doing this work, and I mean, maybe that's something of interest that we can discuss here at some point. We are now using other protocols that do pattern, and I think moving forward, we definitely would want to do that. So for example, Leah is preparing a project where she's going to look at sex differences, and there we'd like to look in pattern protocols. So then this is something we could look into, but it's not something we've focused on.
Great, thank you.
Christiana, it's really good. It's great to have you present as well. So I was, I don't really understand very much about the work with the organoids. So maybe the questions I'm going to ask you are naive, but so you have the organoids and you treat them with you stimulate them, with the glucorticoids. And then the chronic challenge that you do is for 10 days. Is that right? Did I get that right? Yes. Okay. And then during these 10 days, the challenge is at the same level. So you challenge them equally, putting in the medium the glucorticoids. Is that at the same?
Yeah. So we add to the medium based on the half-life of the compound. So we replenish, I think, on a 2-day schedule for dexamethasone. And so we don't know for certain that the concentration, if that's what you're getting at, that the concentration is, it's not necessarily consistent during this time, but the compound is active. So it's going in and out of the cells.
No, my question was actually, I mean, my thought was a little bit on the other way, because, if you think about, and maybe, Elizabeth can help with that, but, if you think about in real life, yes, so chronic exposure, what you're trying to model, it's basically a situation where people are, are chronically under stress, yes, but the stress is not constant all the time at the same level, I guess, no? So that you have variations in levels of the hormones over time. Is that something that would, did you, I mean, the field has debated or not, or in terms of modeling organoids? And so that was, I guess, my question is that why do you choose 10 days as opposed to, a larger period of time, or using intermittent exposure as opposed to a constant exposure, so that's, that was basically whether this is something you considered. And then the second thing is that when you do the organoids, and then when you sample them, you take the whole organoid and then you homogenate and then you sample, or how do you do it? Because there's a different structure within it. Do you sample in different places? Is that, or is it too small? I mean, I just don't know.
Yeah. Sure. So I can try to answer all those things. So for the first question, it's a very good question. And so, are modeling something, we decided on a format of model. I agree with you. There's so many different ways we could do it. I think intermittent exposures make a lot of sense. So at first we tried to understand, we did a few different kind of paradigms, longer, different concentrations. We haven't really figured it all out. And part of it is also because we don't know exactly what developmental period makes the most sense. So we tried to match it with this. I mean, we tried to map it to, for example, fetal brain in order to understand what would be the right time if you're thinking about the levels of cortisol that are present in utero, and how, when, for example, dexamethasone would be given, things like this. We tried to take these things into account, but I don't think it's perfect. So in an ideal world, I would do all these different paradigms to try to understand. And I think different ones model different things, right? And so I think there's still a lot to be learned there. 10 days was kind of arbitrary, to be honest. It made sense between the time frames that we knew the organoids are sort of mature enough, but not too mature that it's hard for us to work with them. So some of these choices were in fact practical.
Yeah, maybe Christiana, maybe I can chime in. So this was basically, we had this one paper where we looked at human hippocampal progenitor cell line. And there also we sort of followed a protocol that was established by King's College, where they had actually treated cells for 10 days. And then there was a washout period, and they treated the cells either while they were differentiating or after differentiation. And there we saw some lasting effects on DNA methylation that were leading to sort of a priming of a then enhanced transcriptional response to glucocorticoids. But in the model that we had in the cell line, it was quite diverse, and we didn't know, are we looking at progenitors, neuron, and there's even glia in there. And so we really wanted to, I mean, the easy sort of explanation is we wanted to sort of do the same experimental protocol in the organoids to test that hypothesis, but then on a single cell resolution. Mm-hmm. And of course, dexamethasone has an advantage. It's quite specific for the GR, but it binds there and it sticks there. And it's a pharmacological agent. And having hydrocortisone, for example, is something that we're exploring right now also, because it's more of an on and off also at the receptor level. And the other thing that I'm not sure Antti also wanted to say something is that we just also don't know what the exposure in utero is. Like, what do you see circadian fluctuations in the baby, or how is the placenta controlling that? Yeah.
And I also wanted to note here that the organoids are human neurodevelopment, and human neurodevelopment takes longer by default from the developmental time viewpoint than mouse, for example. So if we want to study the effect of any pharmacological effect, now the dexamethasone, on the differentiation of our neurons, we would need, for example, I've tested that 7 days to see a progenitor becoming a neuron. In the mouse, the same time period is 2 days, for example. So the 10 days is a good time period for the cells that we affected 10 days before that they have become what they would become, and see all the in between. So 10 days in the organoids makes sense.
Okay.
And so for your second question, whether we dissect them or not, so this we started out doing in the beginning, that's what we were doing, trying to dissect the ventricle. So these organoids, I showed you pictures of these ventricles, which are the functional unit, and they're mostly the cells we're interested in, but they have other structures that are not as interesting. And when we've done the single cell experiment, from a practical point, so before we were using a technology that allowed us to maybe sample 2,000 cells. And then when we switched to 10x genomics, where we could get a lot more cells, we decided to go with just associating the full organoids, because the single cell data will allow us to focus on the cell types of interest. And this allows us to have much higher quality data because there's less processing. So we could dissect, but it's not worth it in the quality of data you get, and you could misinterpret things because of apoptosis that you're inflicting based on this elaborate process. So yeah, but when we're trying to validate things, for example with staining, then we're able to look at the structures that are most relevant.
Okay, so you basically dissociate all the cells and then you run them. Is that
exactly?
Yeah.
Yeah, and what to account for heterogeneity, which is true of these things, of these organoids, then we need to combine multiple organoids in a particular N, and we have to do multiple N so that we can correct some of these differences, which when you're looking at primary tissue, it's a lot less of an issue, right? You have more consistency. Yeah.
Okay. Very good.
Okay, I guess if there's no more urgent question, we should sort of close the session because we're anyways a bit over time. But I think it also shows that this is, it's already very, I thought it was a great, great session, very, very inspiring presentations and discussions.
Absolutely. I'm looking forward for the next one, which is Jermaine, June
22nd of June.
22nd of June.
Yeah, we'll send out more information to come.
All right. Okay. Well, thank you very much, everyone. Have a good one.
Okay. Thank you. Bye.
Bye.